Okay, these are your notes for lesson 2.1 on the scientific method. In this video, you will take notes on the general scientific method process and also notes on accuracy versus precision. You will follow along with your doodle notes. There are blanks where there'll be highlighted words in the presentation that you'll fill in and take any additional notes or activities that the doodle notes will tell you to do. So we first want to know what the scientific method is. In general, it's a map or a procedure that is used to discover the world around us. We can draw conclusions for the way things work and behave in our world using the scientific method. In general, we want to ask questions, we want to define problems, do background research, hypothesize, experiment, analyze, and conclude our information. Now, sometimes the process doesn't work smoothly and we need to start over, and that's okay. The idea is to ask questions and to move to figure out how the world works. So, on the next couple of slides, you're going to see each step with a little blurb to identify what that step means to you. Write as much or as little as you want for you to understand the step. So, the first step is to make observations. Once you make observations using your five senses, we're going to define a problem. When we define a problem, you're really asking questions that you wonder about or that you're interested in. When we pick out topics that we want to do, we want to really follow our curiosity and want to figure out what is exciting and what's interesting that we want to know. That's what makes a good project. After you come up with your topic, we want to do our research. Our research is going to be any background knowledge that is already known about your topic. If you're doing a project on football, you want to know everything that there is about football. If you're doing a project about plants, you want to know everything there is about plants, how much water, soil, what kinds. You want to know everything you can on your topic, whether you're going to use it or not. When you do research, you want to make sure that you use reliable and credible sources. Things like .edu, .org, or .gov are really reliable, good sources. Sites like Wikipedia and Googling it are not. Once you come up with your research, then it is time to plan. When we plan, you're going to come up with your variables, what you're changing, what you're measuring, what you're keeping the same, and you're also going to write your step-by-step procedure of how you're going to test your question. Of course, no plan is complete without our hypothesis. A hypothesis is your educated guess. It's an answer or a possible answer to your question, what you think is going to happen. It's a prediction. A hypothesis is generally stated in if, then, because format, and we'll talk more about that in our next video. Once we've come up with our hypothesis, your plan, and your variables, then it is time to experiment. You're going to use your procedure and walk through it step by step with details and changing anything that you need to as you go along. You want to do several trials in your experiment to make sure that you have reliable sources, or not resources, reliable data. As you're running your experiment, you will be collecting data. You usually collect this in data tables, but there are two types of data that you're going to be using. We have our observations, which are called qualitative data, and then you have your measurements or your numbers, which is called quantitative data. You need both of these to make a reliable conclusion to your data. I've actually done this video several times. My computer wasn't working. Now I'm getting a little tongue-tied. I apologize. Once you've collected your data, both qualitative and quantitative, you're going to analyze your data. That's where you put it in graphs and charts, and you discover any trends, any outlying data, any surprising data that you've noticed, and you're really going to be looking at were you right or wrong. And finally, we are going to make a conclusion. In your conclusion, this is where you're going to either accept or reject your hypothesis. If you are right, then you're going to accept it. If you are wrong, you're going to reject it. Sometimes you might be inconclusive. You might be partially right, but you might not have enough data to support fully what your guess was. So you might be inconclusive. And that's okay. You don't have to be right. You don't have to be wrong. You can go through the process again to have a better understanding of that problem. And then, of course, you can also report your results. For us, that's going to be your science fair formal report. You will report your results to me, who then will report them to the school fair, the county fair, and then maybe even the state fair later on. If you're serious about going through with all the steps, then you could test it again and again and again to eventually become a theory, and eventually theories might even become a law. So, not all experiments, let me move my face out of the way, not all experiments can become theories, and not all theories can become law. It takes many, many years and many, many scientists to figure out whether something is a theory or not. And then, of course, we want to talk about what makes a testable question. So, as you are going to be introduced to science fair later this week, you really want to come up with something that is testable. Testable questions have a specific manipulated and responding variable, one thing that's changed, one thing that's measured. Typically, a testable question is going to be a how or a will or an if question. How does water affect plant growth? Will changing the amount of water cause plants to grow taller? Will water increase or decrease the plant height if changed? If water is increased, then the effect on height will be increased. So, those are going to be testable questions, things that you can test. An untestable question is a why question. You're not really indicating variables. You don't really have anything to measure. And it's a question that you could Google. Why do blackberry bushes have thorns? However, we could turn your why untestable question into a testable question. How do the number of thorns affect how many animals eat from a blackberry bush? So, no matter what kind of question you come up with, we can test it, turn it into a testable question that you can create a plan for. The last thing in our notes for today are going to be how we make an experiment fair and valid. And we've already talked about testing several times, making several trials, and that's going to be to going with our precision and our accuracy. Precision is how close a group of measurements are to each other, whereas accuracy is how close the measurement is to the true or accepted value. In science, these are important to make an experiment fair and valid. We want to test our experiment several times and make several trials because we want to be precise. The only way to be precise is to test it several times. If you test it several trials and your results are coming out around the same every single time, then you're precise. However, in science, we also want to be accurate. We want to be close to the accepted value of whatever we're trying to figure out. So we want to be both precise and accurate in science. To really understand precision and accuracy, we use targets. And this would be darts, bow and arrows, anything that uses the bullseye. In this first diagram, we have three arrows that are hitting around the middle, but they're not in the same spot. But we're in the bullseye. So we say we're accurate, but we're not precise. They're not exactly the same. In the second diagram, all three arrows are hitting the same spot. We're precise, but we're not in the middle, so we're not accurate. In this third one, all three arrows are in the exact same spot and they're all in the bullseye. We're both accurate and precise. And in this last one, well, I think you can probably see that that's neither accurate or precise because they're all over the place. We can use this to talk about how our trials are close together in measurements and how close they are to the accepted value using our research. We need both precision and accuracy to make our experiment fair, valid, and credible.